All right, in the last section we talked about carbohydrates. In this section we're going to talk about proteins. And we said proteins are a large macromolecule that consists of many monomers, many amino acid monomers that get linked together through these dehydration reactions. One of the things that we did in class when we started this was I asked you to take two generic amino acid structures and then link them together. And we did that. And in the process we gave off water. We took the amino group of one amino acid and joined it with the carboxyl group of another amino acid to get this chain formed. And in the process, we formed a peptide bond, and water, of course, was given off. And in this process, we said in our bodies, as our bodies grow and repair themselves, this process is run by enzymes. Okay? So proteins are polymers of amino acids, of course, and within our bodies, they're the same 20 amino acids are used to make all the proteins within our body. And they consist of one or more specific peptide chains that can be linked together to give the actual functional protein. So you can have one peptide chain that gets linked together, and grows and forms a functional protein, or you can have multiple subunits that get linked together to form the protein. And we talked a little bit about that, and I'll go into a little bit more detail on that here in a couple minutes. Okay, so if you look at the protein here, or the amino acid, I should say here, you've got the generic structure, and you've got the amino group, you've got the carboxyl group, and those are two of the seven functional groups that I asked you guys to write down and know. And then to that central carbon atom, the pink one here, this is where you have the radical, or the R group. And that R group is what makes each of the amino acids different. Okay? And so the proteins, when we link the two amino acids together, or as we continually add an amino acid to this growing peptide, what we get are these dehydration reactions, which links, again, of course, the amino group of one amino acid to the carboxyl group of another, yielding water and forming the peptide bond. So here it is again, the same picture we looked at earlier, and this is something that you want to make sure that you're able to do, which is why I'm kind of reiterating it and going over it again and again with you. Okay? So if you look at these amino acids, as they begin to get added to the growing peptide chain, they begin to interact with one another. And in doing so, what's happening is this protein starts to take on its three-dimensional shape, or its three-dimensional conformation. It starts to fold and bend around as all of these things start to interact with one another. And an enzyme is an example of something that has a particular shape, and when it interacts with a particular substrate, that shape of that enzyme, which is determined by all of the amino acids in there, that determines how it's going to interact with that substrate. And then we talked about 4 different levels of protein structure: the primary, secondary, tertiary, and quaternary. So for instance, when you're looking right here, it looks like 1 degree, 2 degree, 3 degree, and 4 degree. That's just kind of a shorthand notation way of writing primary, secondary, tertiary, and quaternary. Okay, so the primary structure of the protein is just the order of the amino acids. That's all it is. Nothing more, nothing less, just the order of the amino acids. Okay, and when you look at that primary structure, it begins to, as the amino acids are being laid down, they begin to interact with one another, and in doing so gives rise to the secondary structure. So in other words, one of the things that we looked at in our textbook is the fact that there's polar and nonpolar, and then there's electrically charged amino acids. And due to these differences in the chemistry of them, we talked about how they start to interact, and they can potentially hydrogen bond to one another. So these hydrogen bonds, as a result of these interactions, is what's giving rise to the secondary structure. And the two examples that we talked about were the alpha helix and the beta pleated sheet. So when you think of primary structure, you should be thinking of the order of the amino acids. When you're thinking about the secondary structure, you should think about hydrogen bonding, and that gives rise to the alpha helix and the beta pleated sheet. Okay, so when you talk about the secondary structure, you can just go ahead and take a look here. These are the two things that I just mentioned with the alpha helix and the beta pleated sheet, of course. Now with the tertiary structure, this has to do with those R groups. Okay, so what's in that box that has the R in it that makes all of the amino acids different? As these things begin to interact with one another, they too are contributing to the shape of the actual protein. Okay, and the R group interaction gives rise to the tertiary structure. And you can have some hydrophobic interactions, you can have some hydrophilic interactions, you can have some, electrical interactions, those different types of things with the different R groups. That's what's going to give rise to this tertiary structure. Okay, now we have, if we look here at the quaternary structure now, The quaternary structure, if the protein consists of 2 or more subunits, then that would give rise to the functional protein. So with the tertiary structure, if the protein itself is functional at that level and it doesn't consist of any more subunits that are necessary, then that tertiary structure can be the functional protein as well. With the quaternary structure, if we have multiple polypeptide chains that need to interact to give rise to a functional protein, then that would be the quaternary structure. So for instance, the examples that we gave in class were collagen and the hemoglobin. And we talked about collagen being one of the proteins that's in your skin that kind of gives it its structural integrity, the resiliency, prevents against like mechanical strain and that sort of a thing. And it's got 3 kind of subunits that get woven together almost like a rope, as you can see here in this middle structure. And then if you look at hemoglobin, we talked about this being responsible for carrying oxygen to the respiring tissues and carrying CO2 away from the respiring tissues back to the lungs. And it consists of 2 alpha chains and 2 beta chains. So 2 alpha globin, 2 beta globin that get put together to make the final functional protein. So again, this is a case where there's 2 or more polypeptide structures that are linked together to form the functional protein. And that would be, of course, the quaternary structure. Okay? So now as far as your proteome goes, we talked about your proteome in class. Just like your genome is a collection of all the genes, your proteome is a collection of all of the proteins in your body. And of course, proteins, we said, are coded by genes. So you have these idea of these triplets, and these triplets code for the particular amino acid that comes in, and then that amino acid gets added according to what the triplet says on the messenger RNA that was made from the triplet code that came from the DNA. Each individual has your own set of genes, of course, and those little subtle differences within those genes is what makes us all look a little bit different. And you can see the differences in your genes are what's going to give rise to the different proteins in your body. So, of course, just like your genome is unique to you, your proteome is unique to you as well. We all code for the same proteins, but some of them are slightly different, and those minor differences, of course, are what make us all different. So now as far as the genetics goes, one of the things that we can look at is in normal hemoglobin here, what you have is glutamic acid at that 6th position. So if you look at the very top of that figure there where it says normal hemoglobin, and it's numbered 1, 2, 3, 4, 5, 6, 7. If you look at the 6th spot, it says GLU, and that's glutamic acid. In normal hemoglobin, glutamic acid is added in, and your blood cells look like they do here on the bottom left. The red blood cell shape kind of almost looks like a jelly donut there, slightly concave in the middle and so forth, and it's rounded, nice and smooth. Well, with sickle cell anemia, if we have one error, for instance, in that 6th position, you can see on the right there where sickle cell hemoglobin is valine, gets substituted in instead of glutamic acid. So there's one difference in the letters, the A, T, C, and G. There's one difference in the letter, and it codes for valine instead of glutamic acid. And what that results in is a conformational shape change in the actual protein in that beta globin molecule. So instead of having this nice rounded smooth hemoglobin, smooth red blood cell, it causes the red blood cell to actually change its shape. So that conformational change, that quaternary structural change in the hemoglobin has a dramatic impact on the red blood cell. If you look at the bottom right there, you can see it's almost got that sickled shape. It starts to get bent. It's got sharp edges. It's not very smooth and round, so it tends to get caught in the capillary beds in your body and causes a whole host of problems, which we'll talk about as we move on throughout the course. We'll talk about these things, but you can see that one change in the genetics leads to a humongous change in the actual structure of the protein, and that leads to a big problem. Okay, so that takes care of the protein parts. Of course, as always, if you have any questions, please let me know. You can see that I had skipped over a couple of videos here that we're going to make sure that we watch in class as well. Okay, so we'll talk to you soon.